I apologize for interrupting. This is Dallas. Could y'all unmute so we could hear?
Can you hear now?
20 minutes in.
Yes, thank you.
I might just start over.
This is this shows the other extreme, and you can actually see the stigma quite well here. The pollen grains have terminated, tubes are growing in and going down through the stigma and style and on into the ovary. And so this is, you can see the extremes on this. You don't have any incompatibilities here. Now, as I indicated, apomixis is prevalent within many tropical warm season grasses. You might say, what is apomixis? Several of you put your hands up, and you have a good number of you know what it is. It's an asexual or vegetative stage method of reproduction in which the embryo in the embryo sac is formed without fertilization of the gametes. In other words, seeds are produced without the union of the egg cell in the embryo sac and the sperm nucleus coming from the pollen. You have, as far as apomictic plants, you have two types classified. You have obligate apomix. In this case, all seed are formed by apomixis. And one plant that I'm aware of that I would say is an obligate is common Dallas grass. It isn't all that common. Most of them are facultative apomix in that the seed are formed from both sexual and apomictic reproduction. And this is obviously the most common in grasses. Some of the effects of apomictic reproduction is meiosis in the developing of the embryo sac. Meiosis or products of meiosis are bypassed in the ovule. The embryo sacs develop from an unreduced 2N somatic nucellar cells in the ovule. And an unreduced embryo develops directly from an unreduced egg in the embryo sac. And here, this is called parthenogenesis. It's the embryo, the egg cell just starts developing on its own, and you get a cell that's embryo that's exactly that develops like from the genetically it's similar to the female parent. And it produces pollination is required for endosperm development. This is called pseudogamy. And this endosperm is going to be, of course, in your seed. It produces uniform progeny that are identical to the female parent. This is very, very important.
Byron?
Yes.
On the common Dallas grass, is there a way to get over the obligate apomixis so you can make crosses?
No. Well, you can use it as a male parent.
You can use a male parent. Okay.
I spent almost a whole career trying to get around that. Another story. Classification of apomictic plants. Did I go backwards?
Yeah.
Well, the mechanisms, we have two types in grasses. Apospry is an unreduced embryo sac that develops from an ovule, from ovule tissue other than the megaspore mother cell. And this is most common in warm season grasses. And then we have another one called diplospry, that the unreduced embryo sac originates from the megaspore mother cell. And this is a cell that normally undergoes meiosis, but meiosis doesn't occur. And this may be a little bit of an overkill for you, but is what I'll be going into. But let's go back first. Let's get a common place here. Let's go back to sexual reproduction, and this is Botany 101. Most of you have forgotten that. I've drawn what is a ovule in the integuments, and this is inside the ovary of the developing flower. And you've got your nucellar tissue inside the ovule, and there's one cell, the megaspore mother cell, enlarges and undergoes meiosis. So if this has got a chromosome number of 40, you form a dyad and then a linear tetrad. Each of these are N in chromosome number, and you have chromosome number 20. And you've had genetic recombination taking place and crossing over. The lower three of these degenerate, and the one furthest from the micropyle starts enlarging, and it's called a functional megaspore. There you can see the remains of the three that degenerated. And then it divides mitotically to produce 2, 4, 8 nuclei, and these differentiate as egg cell and the two synergids. Everything, all these are reduced in chromosome number to N or 20, and then you have some antipodals up here, and they usually proliferate. Now, this is sexual reproduction, what's happening with wheat and most of your crops you're breeding. This is a section with a microtome thin section through an over in an ovule showing an actual sexual embryo sac. Here's your egg cell. Don't see the synergids, but your polar nuclei and your antipodals. Of course, a sperm nucleus is going to fertilize the reduced egg cell to give you get your chromosome number back up to 2N, and then 1 will fertilize your polar nuclei to form your endosperm. Yeah. Need you over here. Okay, now we'll go to apospry, the most common form. Back to our drawings again. In a lot of apomictic plants, it will undergo meiosis, the megaspore mother cell. This is identical to what I had in the last one, and you have the functional megaspore. But at about this time, the nucellar cells near the functional megaspore will start enlarging, and some of these will start undergoing mitosis, and at the same time, the functional megaspore will degenerate. And so you end up going through mitotic division. Now remember, these are 2N in chromosome number, and so these nuclei are 2N. And they'll differentiate into sacs that have, here's a buffalo grass type, you have 2 synergids, egg, and 1 polar. And then you can find them with 1 synergid, egg cell, 2 polars. So you have a lot of variation, but everything in there is unreduced. So chromosome number 40, those nuclei have got 40. And here's it doesn't show up too well, a section showing you have one nucellar apomictic aposporous sac here, and you can't see the nuclei in this section too well, and then another one here. So you've got anywhere from 1 to 4 or 5 sacs in an aposporous ovule. The other form, diplospry, as I said earlier, the megaspore mother cell will enlarge, but rather than undergoing meiosis, it just enlarges, and then it starts dividing mitotically to produce an embryo sac. So this is extremely difficult to detect, because if you remember the sexual embryo sac, we had antipodals and everything else. You got your polars, your eggs, synergids. It looks at this stage just like a reduced embryo sac in a sexual plant. So you really got to observe what's going on back here in the early stages cytologically to pick that out. And here's a sac that looks like one. It's not very good, but here you see the antipodal, polar nuclei, and egg down here. Well, okay, you have variation in your embryo sac development in the facultative apomix. And if you remember, I said the facultative apomix are the most common, and you can have any of these scenarios, or all of these taking place. You can find ovules that have a sexual embryo sac, and this is in an aposporous facultative apomix, or ovules with only apomictic aposporous sacs, as shown here, or you can have an aposporous sac here and one here, and then you have a sexual sac with antipodals here. So you have this occurring, I've seen this occur in the same inflorescence of the plant. So there's some influences of the environment, obviously, or other factors involved. Just to show you actually a ovule that has this in a facultative, you have one sac that comes in right in through here. You have the polar nuclei, and these cells right in here, and they'll show up on the next one a little clearer. This is a pro-embryo. The egg cell has already started dividing on its own. Whereas over here, you have your antipodals, you have a sexual sac, egg, and the two polars. Now the next section through this ovule, you can see a little more of the pro-embryo. You can see it's developing here, and here's polar, and here in the antipodals of this sac here. Now since this pro-embryo is developing before this egg cell up here has been fertilized with a sperm cell, this is probably going to develop into an apomictic plant and just crush this one out because it's developing earlier. Okay, the outcome of apomictic reproduction, going back to this, said that you have uniform progeny that are identical to the female parent. And this is one way to pick up apomixis in the field. These are bufflegrass plants that show these are different accessions. There's a division point right through here. You can see these inflorescences are different from here to about here, and then they're different back there. But you can see this color change, different from the one here. Here's another accession. And so you can see the uniformity that apomixis does give you, in that you're just cloning whatever the female parent was. So if you're out here trying to cross that with something else, you're just wasting your time if you pick out one of these and do it. So let's go now to a brief overview of breeding apomictic grasses. Improvement of, this goes back to your question, Seth, improvement of obligate apomix, where there's no sexual types are available to cross with your apomix. The only way that you can get a better grass is to go into nature and try to select a superior apomictic ecotype that's growing in the wild. A good example of this is in bufflegrass. Common bufflegrass was selected out of South Africa back in the 40s, and Soil Conservation Service released it, and it's probably even still today the most common cultivar that's grown in South Texas and in Mexico. Someone had a good eye and they picked out a very good natural ecotype and released it, and Mother Nature sometimes develops some very good material out in the wild. Another way you can get a little variability in it is fertilization of an unreduced egg. And you said, wait a minute, you said that the unreduced egg's not fertilized. Occasionally, pollen will light on the stigma. Remember I said they had to be pollinated for the endosperm to develop. And the pollen tube will grow down before the egg cell starts developing into an embryo. And you can get fertilization. And that's called B3 hybridization. Let's say the female parent here had 40 chromosomes. Obviously the egg is unreduced. And then a 20 chromosome reduced pollen grain lights on the stigma, and the sperm has 20 chromosomes, well, you're going to end up with a 60 chromosome plant. And sometimes you can bring in variation through that pollen that it's undergone genetic recombination, and so you can get some different types with this occurring, but it occurs at a very, very low frequency, and it's not guaranteed they're going to be any better or as good. Trying to induce mutations using mutagenic agents and radiation has never been successful, and some somoclonal variation through tissue culture was tried and to no avail. So basically this is the only way you can try to improve a completely obligate apomictic biotype or something of that nature. Now, apomixis is a valuable breeding tool if sexual germplasm is available. has to happen though, if you're gonna make any improvement, the sexual plant must be cross-compatible with the apomictic male parent. And you make the cross using the sexual as a female and the apomic as the male parent. Apomics produce good pollen or reduced pollen. All this on apomixes is all occurring on the female side. So you take your pollen from your apomic, put it on your sexual plant, and you're bringing in genes from your apomic, and you've had genetic recombination, and so you get a release here in the progeny F1 hybrids from this. And for this to work, your F1 hybrids should segregate method reproduction and other traits, of course. And as I just indicated, most apomictic plants are highly heterozygous because they're of hybrid nature, a lot of them are. And the F1 hybrids, you have a wide array of types. It's a release of the genetic variability that's stored in that apomictic plant and not expressed. And it's amazing what you can actually get out of a cross between a sexual and apomic. All traits expressed in the F1 hybrid, including heterosis, if there is any, are permanently fixed if that F1 plant is highly apomictic. And forage grasses, I said, most grasses are facultatives. Now, if you've got something that's 95% or higher apomictic, you're not going to get many off types. And in forage grasses, you don't have to have a uniform product. And usually the sexuals are going to be weaker and they'll be lost anyhow. And if the F1 hybrids are superior to the cultivars that are existing, these existing cultivars, these apomictic hybrids can be released as new cultivar in a short period of time, and I'll go into that when I talk about a little bit on the breeding scheme. This is a approach to go, and what you do, you try to find as high of a sexual plant that still has a lot of good agronomic traits, that's highly sexual, 95% or higher. Use it as your female, and a highly apomictic plant with desirable traits. Cross the two. Usually the problem here is the sexual plant doesn't have the traits that you really want, but cross the two, and you're going to get in your F1 hybrids types that are highly sexual all the way across to highly apomic, and then intermediates in here. And they're going to be also segregating for plant height, for other traits, because as I said, you've got a lot of heterozygosity stored at apomic. Now, if these are potential hybrids, if you find something here that you want that has good traits better than anything you think what you have in other cultivars that are available, get the seeds from this, put them in a evaluation test, replicated at a number of locations, and put a check in your desirable cultivar that's being used on cultivars. If you're only getting forage production, you can, in 3 years, you know if it's better or not, and if it's adapted, then you're ready to release, rather than going what you go through on a sexually reproducing plant. Now, if none of these are desirable, you can go back and pick out maybe sexuals out of here that might be more vigorous or better than that, or maybe some apomictic lines. Repeat the process, and then you have another population to deal with here, and possibly pick out some from that group of apomics.
Dr. Burson?
Yeah.
How do you quickly screen out between the sexuals and apomictic F1 hybrids?
Uniformity.
The progeny test, progeny row?
Right here, yeah. That helps now, because on your facultatives that are about 50-50, it's almost impossible to know if there's apomixes in there or if it's totally sexual. But you would go in on progeny. You could look at the uniformity within the progeny, yeah. And this, see these plants right through here? These are some sexual segregates out of a cross, whereas these is, it's always hard to show on pictures, but These are uniform. This line over here is uniform, but the sexuals are less vigorous, and that ties into exactly what you were asking. Here are some apomictic hybrids progeny out of it. You can see here you've got the blue color in this row, uniform. I have another one here. These may be 2 rows per hybrid. Look, very little flowering here, blue color. Looks like more forage than what you have here. And these are flowering and a lot of seed heads. And over here are some more blues, and you can see in the background different types that are showing up. I would probably bet that there's some sexual types showing up. These are highly uniform, and this is what you would do. Once again, row of highly apomictic types. You see some off types showing up in this row, but some more apomixes here, and these are very good, vigorous forage plants. And these would go into a test to come up with your, see how well they're adapted to the out yield, what's available. Now, program here at College Station has released a number, well, there's 2 that my predecessor released that were developed this way, Nueces and Lano buffalo grasses. And with that, we'll stop for any questions.
So how many progeny would you plant to screen for that?
I like to use about 20 or more
Rows or 20?
plants per
Oh.
entry. You could go higher, but with 20, you will find out how much real quickly if it's highly, how much sexuality and well, you're interested in how apomictic it is and if there's any off types. If you don't find 20 if you don't find an off type in 20 plants, you're going to have a highly apomictic type.
Byron, you said they were the apomix are highly heterozygous. Is there like a range of what is typically seen as far as the level of heterozygosity?
You gotta remember, we're working with forage grasses, and I don't know of anyone who has, other than just looking at the pictures, that there may be some in the literature, but we're not working with these
Right.
domesticated crops, but it's amazing how much you can do with, how much is just the variability expressed in that hybrids when you have a sexual type. Well,
Byron? Oh, sorry.
My question is, this relates to apomixis, because you've got it, a lot of people want it in other species, and people seem to get confused on whether it exists or doesn't exist. I know it's a little bit off the track, but, you know, like in sorghum, what exactly is your proof in a species like that you do have apomixis?
well, my predecessor, E.C. Shaw and then Keith Schertz, they determined it by, there was, Wayne Hanna picked it up in the field, and to this day, I still don't know for sure what he saw. That there was something that threw a red flag up to it. They brought the material into the lab, embedded and sectioned the material and looked at it, and yes, they were apomictic sacs. But what he's referring to, they never could get the apomixis up, the expression of apomixis up high enough to use it as a breeding tool in sorghum. I think about 30% is about the highest I've ever heard. I know there's work done on it in India, in China, and also here in the United States. But
So I guess my question is, you don't have any idea what he saw in the field, though?
I'll ask him sometime. No, I've asked him and I've probably forgotten. I obviously have forgotten. Yeah. Bibi?
Two small questions. On the
One at a time, because I'm
looking at the segregation, is there any pattern for genetic, whether it is genetically controlled? How many
Oh, yeah,
genes
there's
will?
been a ton of information on that, and a lot of bad models put out. Some have said, well, it's single gene, and then it was 2 genes and epistasis. That didn't fit. And then when they started doing molecular work with it, they found it's a lot more complicated than just 1 gene. And we still don't know the genetic control of it, for sure.
Can I follow up the second question?
Yeah, now you go.
In late 90s, there was a lot of excitement from May's group in Summit and also I think Cornell University that we have the best answer now through apomictic to pick the hybrids. And as if this was the real revolution in plant breeding. And then all of a sudden we stopped reading anything about it. So what happened?
They didn't get what they thought they had.
No, they didn't have a
Now,
process.
the French, if you remember the group out of France, Yves Sauvadon was heading up that group, went to the Summit, made a good collection. This was on teosinte, or not teosinte, trypsicum, dactyloides, and other species, and that's a wild relative of corn. They made a collection because apomixis, it's actually diplospora that occurs in and it's called eastern gamagrass, what we call trypsicum dactyloides. They thought they could transfer the genes for apomixis from eastern gamagrass in corn. And they had a very good program, and they went at it from a more classical, what I gave here, a cytogenetic and breeding approach, but they were doing molecular work at the same time. And obviously they never got, they got it over into some, but you'd have little ears, little cobs about like that. This is all I ever saw. I was working on, we were trying a different approach before I came to College Station, but it came obvious to me I didn't have the resources, money to do it, because the French were pouring millions of dollars into that program that was going in Mexico City.
Byron, you talked about the CIMMYT program on apomixis. Ultimately, it takes a lot of resources to identify genes or even clone genes and use those in transgenic technology to move the gene into other species or within the species. But I know that the private sector is not interested because the producer will just
Use the same seed over.
use the same seed over and over again. And
They
so
were interested.
the private sector was interested in
Yeah,
that.
wanted to get it and keep it. Keep it off of the market.
Shelf.
Yeah, put it on the shelf. Go ahead, Mary, you more?
No. I mean, you talked to Bibi about the fact that some papers showed monogenic control, some showed epistasis. But if that gene can be cloned, do you think it can be utilized in breeding, maybe wheat or wheat breeding or
Genes.
support?
Well,
Gene. I mean,
He said genes.
the trait itself. It can.
Russ did his PhD dissertation, graduate work on mapping buffalo grass for the apomixis genes, and there was some work on some other Pennisetum species going on at Tifton, Georgia at that time. Y'all didn't exactly agree on all the terminology in detail, but explain what you found.
It's in heterochromatic regions, so it's a very large region of suppressor combinations. So a lot of genes can be in there, and their expression is modified. And there are other areas in the genome, at least 2, in the Pennisetums, that affect how it is expressed. So there are some modifying loci as well. So one primary locus, but who knows how many genes are in it. They've done a better job of dissecting it in other taxons.
Yeah, probably known better in buffalo grass and the other Pennisetum species.
In Arabidopsis, they think there are 2 genes that are involved, one around avoiding meiosis and one around partition. That's about as far as they've gotten.
Let me mention one other thing. Apomixis has only, as far as I know, it's only been expressed in polyploids.
In what?
Polyploids, triploid or higher.
Did sorghum in a triploid or a tetraploid?
It was a diploid.
That was a diploid.
But, now, wait a minute, what's the base number of sorghum though?
Well,
Is it 5 or 10?
I have a question
Yeah.
about polyploidy, and I wanted to ask you, since you've got these extreme variance in numbers of genomes and ploidy levels, is there a relationship between productivity and ploidy level?
I think so.
What is that relationship?
Breeder I worked with up at Temple, he felt like even for forages, if you went above the hexaploid level in grasses, you weren't going to make much progress down.
So it goes up and then it goes down?
I don't say up, but most of them are at tetraploid level. Now,
Sugar cane?
we're not, well, that's a second. Okay, let me clear up this. You know, I talked about fertilization of unreduced egg. You remember that? Where it said that you got an apomictic. We did some work and found out that early pollination increases that, thinking we could use apomictic plants as male parent. You know, species like, or genus like Pennisetum is protogynous. In other words, the stigmas will come out, and trips go the same way, up to 3 or 4 days before the anthers come out. But you can pollinate as early, and we did get an increase in the amount of B3 or 2N plus N hybrids, fertilization unreduced egg. But it's still some, iffy on that. But in nature, it occurs at a high enough frequency, and that is why you have range of ploidy levels in an apomictic species, because if it apomic's 40, pollen grain lights over here and it's got 20, and so you get, next thing you've got a 60 chromosome plant. Then maybe another one, you've got an 80, and so you keep building them. And then another reason that y'all were able to synthesize why are there so many aneuploids out in nature. The aneuploids are occurring in the apomictic plants, because in an aneuploid, your chromosomes that aren't paired with anything are going to be lost during meiosis in a sexual plant, eventually, 2 or 3 generations. Whereas in an apomic, it's going through the female side, and it's all 2N to 2N to 2N. And so you maintain the aneuploids in nature that way. That's why we found so many aneuploids in that collection of bufflegrass. Yes?
I think I heard you say that the sporophytic incompatibility was the problem for cross-compatibility. As a limiting factor for being able to make enough crosses as you would like to make, because sometimes it was too much sporophytic incompatibility. That when you test you use the apomictic as the male, and it wouldn't accept the pollen often.
Oh, that would be stigmatic. That was incompatibility. This can happen in sexual.
Stigmatic, I called it. Okay.
Yeah, this pollen tube, this pollen grain lights on the stigma. That was between 2 different species, but in the same genus. They germinate, but they wouldn't grow any further.
Right,
And
so
there's an incompatibility between the pollen tube of one species and the stigma of the other species.
So I had a technical question about that.
Yeah.
Did you try, or has it been tried, like a bud pollination like we use in brassicas to get around we found the cross-compatibility, self-incompatibility is active when the flower's open. It's not so active when the flower's immature yet. I wondered if that had been tried and you get a better result or not.
We have a student trying that right now. It's
very difficult. In Pennisetum parthenium, there's a styler constriction, I guess you would call it. And so we were clipping the stigmas below that constriction and pollinating it, but we still haven't had any luck.
But you've gone in like a couple of days before the flower would open, or just curious.
Here again in Pennisetum, you have the protogynous flowering behavior, and those stigmas come out early before the anthers, and so he can just wait for them to come out and do his cutting. But in that particular study, this was between two panicum, several panicum species. We found, this is what I was showing you, Amir, yesterday. We found stigmatic incompatibilities that wouldn't go beyond the stigma. We found in other crosses, we found different genotypes. Styler, it would grow down to the into the style, would not go the ovary, and we had one that would grow the ovary and lose all sense of orientation, and the polytubes would just go around and around and never go on down to the micropyle. Did you have a question? Okay.
So, on your obligate apomictic plants, I would think they'd be an evolutionary dead end because they'd accumulate mutations and...
Yep.
So, has anybody gone back and looked at those ones that were collected to see if they still exist in their native range, or do they continue to evolve and be lost as they can't compete with their...
Well, they can, the one that I call an apomic and is, for sure, is common Dallas grass. It's throughout southeastern, it's around the world, and it's a pentaploid hybrid. It's a trihybrid. Has 3 different sets of genomes in it. You'll see mutant types. It has 2 things that I've seen in nature that it will come up with where mutations occur. Usually it has purple anthers, and every now and then you'll find one of the yellow anthers. And then pubescence is another thing that will come up. We took that through tissue culture, trying to get some clone variation, and we got different types, but they were usually sorrier than what common was. And what we were trying to do on that was get around. It's very susceptible to ergot, claviceps, paspali. We were trying to get around that and never did it. I did a genetic study on it and tried to identify the progenitors and had some success and some didn't. But at one time, 30 or 40 years ago, we thought obligate apomixis was more prevalent than what it really is.
I would like to introduce Dr. Rao. Can you please? He is a visiting scientist with Dr. David Stelly, and talking about cytogenetics, he is one of the famous cytogeneticists from India. He is working with Dr. Stelly. Maybe for 3 months he'll be here.
1 month is over, so 2 months are
So I am persuading him to share some of his experiences here if sometimes we have time for this. David Stelly and him can share it, so thank you very much.
I hope this, to the students, I hope this gives you some idea of some of the problems that you can encounter in working with wild species. And I know if you're working with a major crop, you're not going to involve it, but as Dr. Rooney indicated in his seminar, when Dr. Price, George Hodnut started doing the crossing of sorghum bicolor with the wild sorghum species, that's getting into the wild stuff and bringing it in. It was very difficult until he found the IAP allele. And but I hope this gives you some idea of what can happen if you go home and they say, well, we want you to work, the only job you can find is something on a wild species. And I might add that horticultural people, of course, you're quite aware of apomixis that occurs in strawberry, a lot of the berries, and fruit crops in Rosaceae family, is it? And it's quite prevalent. And well, one thing I want to tell you, Mendel tried. There's Heraceum, a genus. He tried to do a genetic study with a Heraceum species that's called hawkweed, and it's closely related. No, it's not closely related, but over near dandelion, and apomixis is prevalent in dandelion. He made crosses. And of course, if you're crossing apomictic species, what was his ratio going to be? And if he hadn't changed from that to P, how much would he be today?
You want a ride share over there?